Good morning and welcome, AP Biology students and teachers. I'm going to share just a couple of slides with you before Ms. Cook begins her presentation. So please just let me know if you can see my screen.
Yeah, I can see it.
Fabulous. Okay, welcome to our first AP Biology content review session. Presenting will be Mrs. MaryBeth Cook at 10 o'clock and myself, Sheri Bonzo, at 11. There's some information in these slides that will be helpful for you in finding documents a little bit later on that you will want to go back and review, and our recordings. So we have this disclaimer. While this online review session is intended for a Kentucky audience, we will have students from several other areas, and we welcome you. It's wonderful to have you. Again, our host is Advanced Kentucky, and you can follow us on Twitter. If you have a Twitter account, I love my Twitter, so please follow us. Our Senior Content Science Director is Lou Akampora. Everyone knows Mr. A. And if we could please remember our meeting etiquette today, keep your microphones muted, your videos off, but please feel free to use the chat often if you have questions or for polling and participation. Again, there will be a recording of today's session, and you can find this in our content review sessions here at bit.ly/advancedkentucky. And we thank you for joining us today. We're excited to have you and look forward to learning a little bit about biochemistry and membranes today. So with that said, I'm going to stop sharing my screen and send this to Mrs. MaryBeth Cook.
All right, can you see that okay, Sheri?
Yes, ma'am.
All right. So good morning. Thanks for joining us on a Saturday. Today, we are going to talk about biochemistry, and we're really going to focus on the structure and function of these macromolecules. I'm going to start out with some kind of generic information, and then we'll get into the specifics of each one and kind of give you guys a chance to tell me what you know rather than just seeing what I know, okay? So I think it is important to kind of tie this into the bigger picture. So, why do we look at these macromolecules? We know that living things are exchanging matter with their environment, and they need to do that so that they can grow, they can reproduce, and it helps them in maintaining their organization. So when we look at these macromolecules, carbon is that main element in living things that we'll focus on. And I think it is really important if you're seeing this word, organic molecules, in your mind, you should be thinking, okay, that molecule has carbon and it has hydrogen. And more than likely, if they're mentioning organic molecule on the test, then you should also be thinking of these 4 specific macromolecules. So carbohydrates, lipids, nucleic acids, and proteins. I hope I have enough time to kind of touch base and talk about all of these, but I will spend more time with proteins and lipids. So why exactly is carbon so special? It is able to form 4 covalent bonds. So if you think back about what a covalent bond is, those are really strong bonds, and atoms are sharing electrons in that bond. And so carbon can bond to itself, it can bond to phosphorus, not nitrogen, hydrogen, oxygen, sulfur, Those are going to be the common elements making up these 4 macromolecules. And so since carbon has the ability to form those 4 covalent bonds, we get a lot of shapes. And so shape is going to determine function, and that's what we really need to be able to recognize and think about for any question for a lot of the content of this class. So we can see chains, we can see ring structures, we can see branched, and so that'll be important in figuring out what can that molecule actually do. So some basic terminology that I think is really important to know, and that we'll get more specific terms once we look into the specific macromolecule categories, is monomer. So when you see that mono prefix, you should be thinking 1. So we'll be looking at 1 unit, 1 smaller unit. And then polymer, poly prefix means many. And so that's going to be a larger molecule. And so when we're looking at these molecules, how can they form and how can we also break them down? And I feel like these, knowing these reactions and being able to explain how they actually occur has become more frequently asked on the test. So a dehydration reaction, you know, kind of think about that. If you're dehydrated, you're thirsty, right? You've lost some water or whatever. And so in a dehydration reaction, we are going to remove water so that we can form a bond and build a larger molecule. So in this case, using this terminology, we would take those smaller monomers and bond them together to create a larger polymer. And then hydrolysis reaction, you know, if I break this word down, hydro in biology can mean usually 1 or 2 things, hydrogen or water. And in this case, it does mean water. And then lysis, you should be familiar with that, is to break or to burst, however you want to think about that. So we're going to break bonds by inserting water so that we can break a larger molecule down into smaller components. And so again, just to use those terms, we're going to break a polymer down into its individual monomer components. So here's just a visual of this. So you're seeing 2 monomers, and we want to add them together. We want to build a larger molecule. And so this monomer is going to provide the OH or hydroxyl group. Monomer 2 will provide the hydrogen. So you put those 2 things together, that's your water. And now we have the 2 monomers linked together by a covalent bond. And so I do want to stress that. I think it's important that you know that covalent bonds are what's holding those monomers together. The other thing that I do feel like comes up is this term, anabolic reaction. So a dehydration reaction, we are forming bonds by removing water, and that is building a larger molecule or an anabolic reaction. And then the reverse, so hydrolysis, we're taking that larger molecule that we saw in the previous picture, and then we're just breaking it back down into those individual monomers by inserting that water back in. So monomer 1 receives the OH group, and then monomer 2 gets the hydrogen. And this can also be referred to as a catabolic reaction. We're taking a larger molecule and breaking it down into smaller components. I don't know about you guys, but anabolic and catabolic, those words, they just don't stick in my head of what they actually mean. So I'm really big about, you know, if I can think of a meaning, or if I know my answer would have the same word as like a letter, like the first letter of my biology term, that helps me remember. And so I had a student come up with, for a catabolic reaction, cannonball. And so I thought that was really great. It helps me remember what's going on. So think about a cannonball swinging into you know, some wall and breaking it down. That's essentially what's going on with the catabolic reaction. So we've got a larger molecule, we're breaking it down into smaller components by inserting water. All right, so let's get you guys thinking this morning. So if we had a polymer that consisted of 10 monomers, how many water molecules would it take to separate all 10? And so I want you guys to use your chat. How many water molecules would it take to separate this polymer into its individual 10 monomers? All right, we got lots of good participation going on, so I'm happy to see that. And it looks like we're all in agreement. So, you know, this is a great application. Do you understand what's going on? And you're doing a little bit of math. And so 10 monomers, however many. So if we said 100 monomers, the number of water molecules is going to be one less. So in this case, you guys are correct in saying 9. So good job with that. And then just to see if you were paying attention, what bond holds monomers together? So go ahead and put your answer in chat for that one too. What kind of bond is holding monomers together? All right, so it looks like we're in agreement again. So good job with going with covalent bonds. And so these bonds can have more specific names. When we start looking at the specific macromolecule, there's going to be one for proteins that I will specifically stress. But in general, I think it is important that you know that they're covalent. So very strong bond holding the monomers together. All right, so here's what I would like you to do. When you see this work, so carbohydrates, before I get into it and kind of stress what I feel like you should know, what do you know? What do you remember? So kind of think about what do carbs look like? What does the molecule look like? What do they do for the cells? Do you know some examples? So anything that kind of pops up in your mind, if you'll just share that in the chat and kind of see what you would think about if you saw that word on a test. All right. So good answers so far. Got our specific name, covalent bond name there. Good job. All right, so looks like you guys are on the right track. We'll go ahead and get started so that we can get into more of these. So some of you did already tell me this. So the specific monomer name for a carbohydrate is a monosaccharide. And you guys gave me these examples. So glucose is a single sugar, fructose is a single sugar. And then we have polymers. I don't know if anyone gave me the specific polymer name, but that's a polysaccharide. And some good examples are glycogen, starch, and cellulose. And so knowing their function is important. So, you know, why would a cell need this molecule? And so you all did give me some of these as well. So glucose, fructose, those are great examples of energy. So glucose is usually the preferred molecule for any type of cell. So bacteria prefer glucose for energy. We usually talk about it, and our cells prefer it as an energy source as well. And then we do have molecules that allow us to store energy for later use. So glycogen is a great example of how we as an animal store energy for later. So we store it in our muscles and our liver. And then starch is a polysaccharide that allow plants to store energy for later use. And then cellulose, I can't remember if you guys gave me this one. So that's going to be the sugar that helps provide the structure to plant cell walls. So sugars or carbohydrates can also provide structure to a cell. And so I did want to just provide this picture where you're seeing the two monosaccharides being joined together by this dehydration reaction, just so you're tying it into a specific type of molecule. And so it is a ring-shaped structure. Several of you told me that. And some of you told me that it is, we're only going to see the carbon, the hydrogen, and the oxygen if we're seeing, some molecule that we haven't seen before. And it will be in a 1 to 2 to 1 ratio. I think that was the last comment that I'm seeing. So if they are showing you some molecule you've never seen before, if it has these characteristics, then you can figure out what it is. And then I did just give you another tip. If the word ends in OSE, so if you're seeing some, strange word that you haven't seen before, then that's a big clue that it probably is a carbohydrate. All right, so I want you to look at this picture. I'm not going to ask you anything just yet. I've got a multiple choice question on the next slide. So I just want you to look at cellulose and look at starch and just kind of take notes, see if there's any differences that you see, any similarities, and then I'll give you my question. So I'll give you a second to just look at it. All right, so this is going to be our first question. Sheri, do you want to try the poll, to launch a poll?
Yes, ma'am. Let's try it.
Okay. So the question here is, cellulose is a polysaccharide that provides structure to plant cell walls. Starch is a polysaccharide that provides a way for plants to store energy for later use. Both polysaccharides consist of just glucose monomers. So how can cellulose and starch provide different functions to the plant? And I'll let you guys read the choices. So you've got A, B, C, and D over here on the right. And it looks like Sheri's got our poll started. So if you guys will choose A, B, C, or D, which one do you think is the best option? So I'll try to give you about 15 more seconds. About a minute and a half is what you'll roughly have on real test day.
Okay, and also keep in mind that your responses are anonymous. We do not know who is responding. We can see a total, but we do not know your answer.
Okay, yeah, I think we're past it. Went fast. So majority of you, excuse me, did go with C, and so C is our correct answer here. And so the monomers of glucose, they were, let me switch. They're just arranged differently. So let me see if I can go backwards. So if you look at cellulose, I can always, this sticks out, this little carbon with 2 hydrogens and OH, that kind of always sticks out to me. So if you look in cellulose, that's at the top. And then for the next glucose, it's on the bottom. So it's kind of flip-flopped. And then for starch, that group, they're all on the top. So same glucose monomers. Let me go back. They're just arranged differently. And so remember, a difference in shape will allow a difference in function. So that's what was really important here to recognize. I think there was just 1 or 2 that went with D. So it is a covalent bond that's holding those monomers together, but it's not just in cellulose. So it's covalent bonds holding the molecules together in starch as well. B is not correct because it talks about ionic bonds holding them together. And then A looks pretty good, but environment is not going to cause them to function. The shape of the molecule allows them to function differently. All right, so I kind of just want to give you all a chance. Does anyone have any questions about just carbohydrates? Okay, so just like we did before, lipids, what comes to your mind? What pops into your head? Do you know what the lipids look like? Do you know how they function for our cells? Do you know specific examples? So if you can throw those into chat, or if you have thought of a question on carbs, either one right now will be fine. All right, good. So we've got function and we've got structure. Good. We've got different types of fats. All right, so I'm seeing some good terms that I would want you to recognize and apply to fats. Good functions. Okay, so we'll go ahead and get started. So there are several different types of lipids that we can think about. The thing that I would stress, so lipids, they are a macromolecule, but they're not technically a polymer because we're not taking the same small unit and connecting it to a very similar or same small unit. But they do have smaller components. And I think somebody did give me this. So a lot of times, not always, depends on the fat that lipid that we're talking about. They do consist of glycerol and a fatty acid. So that's very common to see. I did get this term from you guys, so nonpolar, and or you could think of hydrophobic. So what does that mean if we're saying hydrophobic or nonpolar? So if you guys can use chat, what does this tell us about fats? All right. So it does look like good. Doesn't like water, doesn't interact with water, would repel from water. And so I think on questions, when they're, talking about a molecule and they say it's hydrophobic or it's non-polar, it is important that you're thinking what you've just told me. So it's not going to interact with water or it fears water. So some good examples. We will talk about fats in general. We'll look at some steroids, and then phospholipids will be important as well. And so again, I just kind of wanted to show you that picture of a dehydration synthesis of how we can remove water and actually form a triglyceride. So triglyceride is, in my mind, that's kind of that typical fat picture that's going to store a lot of energy for your cells. So there's that glycerol molecule that's going to provide the OH group, and then the fatty acid chain is going to provide a hydrogen. So that's where we would take water out. So it takes 3 waters to be removed to make that really large triglyceride. And so depending on shape, we have different functions. And so that triglyceride that we saw in that previous picture, it's a great energy storage molecule. So remember, energy stored in those chemical bonds that are formed. And if you go back, if I go back to that picture, look at all those bonds. And so that can provide a lot of energy once your cells are needing that. I think some of you told me insulation. So think about blubber on a well, it cushions. I think you guys told me that. And then I usually just like to mention this term. I don't think it's super important for you to memorize or anything, but we store fat, you know, as that energy storage molecule in cells called adipose. So I feel like it has come up along the way sometimes. And I think it's, you know, the more you recognize or the more you're exposed to, I think the better or more comfortable you're going to feel on questions. So adipose cells are cells that specifically store like these triglycerides for energy use later on. So here's a molecule that Miss Bonzo is probably going to talk more about, but since we're talking about lipids, this is an important one. So phospholipids make up the majority of the cell membrane. And if you compare it to a triglyceride, it does have a lot in common. So we still have that glycerol group right here. We have those fatty acid chains that we saw in the triglyceride. We just have 2 instead of 3. And some of you gave me these terms. So this top chain is saturated and this one is unsaturated. And that just helps provide that little, I always call it a little kink. It's like it's kicking its leg out. And if you think about what we want the membrane to do, we want it to be fluid. We want it to be able to exchange materials with the environment. But then we also want it to be selective. So you kind of have that straight leg so that the phospholipids can, you know, create this barrier, but then you have the kinked leg that prevents it from being too packed together. So there can still be some movement in and out of the cell. And so what's different is we have this phosphate group on the polar head and it has a charge. And we also have a charge down here with an at this end with the nitrogen and that methyl group. And so with these charges, that allows this portion to be polar. So it's going to be the opposite. You guys already told me nonpolar means repels water, dislikes water. And so this portion is going to like water. It's going to interact well with water. And so think about that on your cell membrane. The outside environment of a cell is going to be aqueous and the inside will be aqueous as well. And so those polar regions can interact with the water environments, but it allows this hydrophobic core of the membrane to still make up the interior of the membrane and be selective on what gets to come and go. And so again, I think just knowing that terminology and being able to explain why a specific molecule can move through here and others can't, you would really want to focus on that terminology. And so having both polar, non-polar capabilities allows your membrane to be selectively permeable. All right, so we've got a totally different shape. So steroids are still a lipid. And so here you're going to see 4 fused rings so that the rings all touch. And these are just common ones. So Ms. Bonzo may talk, cholesterol is really important in the cell membrane. It helps maintain the fluidity. And then these are your common hormones that kind of help give secondary sex characteristics to us. So I don't know where you guys are in the curriculum, but cell communication, estrogen and testosterone are usually common examples of cell signaling pathways. So those come up pretty often. All right. So Sheri, if you'll launch a poll again for me, we'll try that again. So a quick question on lipids. So lipids are blank and are blank in water. So which choice, I'm sorry, would you go with to describe lipids? All right, so about 20 more seconds. Did I put that in your all's way? Okay, so lipids are, so you would want to go with non-polar. So that would get you down to two choices if you knew that and remembered that about lipids. So they don't interact well with water, and they would be insoluble. So D was the correct answer there, which the majority of you went with. So good job. All right, so this is going to be kind of the majority of time is on proteins. So if you haven't figured it out by now, proteins, the majority of our year is really spent learning about proteins. How are they made? What do they do for us? And so again, actually, I'll back up. I kind of forgot. Does anyone have questions on lipids before we do move on to proteins? Okay, so now, same as before, what pops into your head when you see the word protein? So again, what's it look like? What's it consist of? What's it do for us? We got good answers. All right, anybody else? Last thoughts? All right. So the monomer here, so specific name for the monomer for a protein is amino acids. A lot of you told me that. So good job. I think that's really important to know and not, get that monomer name. You wouldn't want to, apply that to carbohydrates and that kind of thing. So that's super important. There are 20 amino acids found in nature. And some of you, we'll see if you guys know this. So monomers are held together by a specific covalent bond. And so I told you, fats and carbohydrates have specific names for their covalent bonds as well. But I feel like for proteins, it is more important to know that specific name. So what do we call the covalent bond that holds amino acids together? All right. I'm so happy you guys are participating so well today. So peptide bond is it. Great job. And that's kind of how we get the polymer name. So it's a polypeptide. So many peptide bonds holding those amino acids together. And so here's our typical structure. So again, if they're showing you some weird molecule on a test question, you know, hopefully you're going to recognize, okay, so it's got this amino group, it's got a carboxyl group. It's always going to have a central carbon with a hydrogen, and then that R group is what makes each of those 20 amino acids special and unique and have different characteristics. And you don't have to memorize these. And so you can see the R groups highlighted in all these pictures, and they have different properties. So they can be non-polar. So again, that's why it's important. I feel like it's important to know that term. So these amino acids, that R group's not going to interact very well with water, if at all. The polar side chains, these will interact well with water. And then we have acidic and basic R groups that also have charges and would allow it to interact with the partial charges of water. So I think that's important in understanding how we get those different levels of folding, those different levels of structures that you guys mentioned in your answers. And so again, just to kind of go back to that terminology I started us with, you know, they like these questions of tying in the dehydration or the hydrolysis reaction with the specific macromolecule. So here we're seeing two amino acids. So here's one amino acid with that amino group and carboxyl group, same thing, amino and carboxyl. And so they're going to come together to create a peptide bond between the carboxyl of one and the amino of the other. And so we take water out, and then there's our peptide bond. Oops. So something else that I do want to mention from this slide. So polypeptides can have direction. They do have direction. And so sometimes with questions, they'll mention this, the amino terminus. So they're talking about the amino end. So that's this end. And then the carboxyl terminus would be that. So I just kind of wanted to point that out. If you see it in a question, you're not like, what in the world are they talking about? All right, so you guys mentioned this. So there are 4 levels of protein structure that allows that final protein to have its, you know, kind of true functional shape. It's going to allow it to do its very specific job. And so it is important to, I think, if you're thinking about these structures, what does it actually consist of, and what kind of bond allows that level of structure to occur? I do have this. So my Google slide will be posted for you guys to access if you wish. And you may have done this already, but there's a protein folding activity that you can go to, and it allows you just to manipulate the conditions. So like you might have hydrophobic amino acids or in a watery environment, or you might have, you know, charged, and it just allows you to see how that protein would fold up. So, you know, I was virtual when we covered that, and I do think it helps my students have a better understanding of that concept. So that might be something you want to check out. But the primary structure, that's just going to be that unique sequence or the order of those monomers. So which monomer is attached to which monomer and so forth. And then we've already covered this. So you would want to say those amino acids are held together by peptide bonds. And so let's see if you guys remember a little bit of cell biology. What organelle does the sequence of amino acids get assembled? Where do we produce a protein in the cell? So I'll let you guys use chat again. All right, so it looks like we're in agreement again. So ribosomes, I think that's important for you guys to know the function of that organelle. So good job. So what molecule brings the directions to the ribosome? So let's start with that first question. So what molecule comes to the ribosome with the directions to make that specific sequence of amino acids? And again, some of you may not have covered this, but it looks like you guys know what you're talking about. All right, so RNA is going to be the molecule that can actually come to the ribosome and provide the directions to make the amino acids. And so where does RNA get its directions? What molecule provides directions to RNA so that it can go to the ribosome? All right. And so that is your central dogma. I think somebody mentioned that in their comment. And you will get into more details of this. So if you feel like you didn't know that, maybe you just haven't covered it just yet, that'll come up in Unit 6. So it kind of just depends on where you're at in the year. All right, so once we get that specific sequence of amino acids, now our amino acids can start interacting with one another. And so the secondary structure provides two possible shapes to our protein. And so they can be an alpha helix or beta pleated sheet. And those shapes, and I've got a picture of this after this, so you can kind of see what that looks like. So helical, or I'll kind of describe it as it's pleated, it's kind of like a zigzag. Those shapes are going to be held together by kind of weak hydrogen bonds. And so that occurs between hydrogen and oxygen, hydrogen of the amino group, oxygen of a carboxyl group. And then tertiary structure, this is kind of that helps that one polypeptide chain get into its final shape. And so because the amino acids are now arranged in a helical pattern or a pleated pattern, the R groups, those groups that make each amino acid special, they are now in closer contact and can interact with one another. And, now is it nonpolar? That amino acid is going to want to fold towards the center of the molecule to get away from the watery environment of the cell. If it's polar, then that amino acid is going to be happy to be on the exterior portion of that polypeptide chain. So it's happy to interact with the watery environment of the cell. And so if you were describing this on a free response question, you can mention any of these bonds. So lots of different types of interactions can occur to create this tertiary structure. And then quaternary, this is where we have two or more polypeptide chains that are interacting so that we have a truly functioning protein. And I'll give you some examples of that in just a second. So here's a generic picture of those levels. So primary, there's your pleated sheet, alpha helix, and then now our R groups can interact, and it kind of just looks like a folded, jumbled mess. And then here at the quaternary, they've got it color coded. So the light, that would be one polypeptide chain, and then the dark is a polypeptide chain. So two chains interacting. I like this picture, so it kind of takes you through the same thing, but then very common protein is hemoglobin. So that's the protein that helps carry oxygen in your red blood cells. And so I wanted us to have an opportunity to do some writing as well, or for you to practice that, because I think that's how you get better. And so again, you may not have gotten to mutation. So I haven't in my classroom yet, but mutations are essentially just changes in DNA. And we walked through that. So we know that DNA, we inherit from our parents, that gets converted into RNA through transcription if you have gotten to that point in the year. And then finally, that mRNA would go to the ribosome and, provide the directions to say, okay, we need valine to come in, histidine, and so forth to make those hemoglobin proteins. And so this would be normal, this sequence at the top. And then if there's just one letter in DNA that gets changed, then that's going to cause this change in our amino acid sequence. And so if you look at the final protein shape of a normal hemoglobin. We kind of have this concave, round shaped protein that allows those hemoglobin proteins to carry oxygen efficiently. With this one change, we get what's called sickle cell anemia. And so it's the red blood cells no longer round and concave. It's they call it sickle. Kind of looks like a quarter moon shape sometimes, and it runs risk of clotting up. They're more easily or more likely to clump together and create clots in blood vessels, and they don't carry oxygen as efficiently. So I have a question for you, and we'll take it part by part. I think that'll be easier for us to think about and look at. So this free response, the setup of this is going to be like question number 4 on your test in May. So on question number 4, you can count on A is going to ask you to describe something. B is going to ask you to explain something. C is going to ask you to make a prediction. And D is going to ask you to justify your prediction. So I want us to try this. Let me see how much time we've got. 41. Okay, so I'm going to try to give you maybe about 2 minutes for each part. So let's start with A. I want you to describe the primary structure of a polypeptide chain. And I wanted to give you a little bit more detail. So kind of be sure that you're explaining the bond that occurs between amino acids and how that bond can actually form. So I've given you a little bit of direction there. So can you write that in a sentence, a complete sentence? Describe the primary structure of a polypeptide chain. So type your answers in chat, and then we'll talk about it. So just try A out right now. It's okay to be wrong. So now we're quietening down when it gets to a free response. Just make sure you're writing complete sentences. That would be one thing I would recommend for a written response. All right, so we got a response coming up. All right, you guys are doing good. All right. So most of you, if you're reading those or looking at those, you have done a great job. So you would want to tell me that the primary structure of a polypeptide chain consists of amino acids held together by peptide bonds. Some of you did say covalent. I think that's okay. And that is able to form, those bonds are able to form through a dehydration synthesis. So very good job. Now, if I ask you to explain the difference in how a hydrophilic R group would position itself in a region of a polypeptide chain versus a hydrophobic R group, in determining the tertiary structure. So now take B. How would you explain the difference of a hydrophilic R group positioning itself in that tertiary structure of a polypeptide chain compared to hydrophobic? All right, I know these take longer to respond. All right, you guys are doing a great job. Let me read one more. All right. So go ahead and move on so we can talk about the other parts too. So most of you did a great job in your explanation. So that hydrophilic R group, remember that means it loves water, it wants to interact with water. And so it is going to position itself on the outside of that polypeptide chain so it can interact with the water environment of the cell. And then hydrophobic R groups, they don't like water. And so they're going to try to get away from it by moving to the, folding itself in towards the center of that polypeptide chain. And so most of you said that's a very, very good job. All right. The next part is to make a prediction. And so something that I want to stress to you guys is for my prediction, there is a correct answer, but sometimes on these free response questions, making a prediction, there's not always a right or wrong answer. And so please, I think sometimes you all are so, most of you are perfectionists. You want to be correct. You want to have correct answers, and you kind of reserve yourself and don't just put it out there. And you have to on test day. There's not always a right or wrong answer. So as long as you can make a prediction of what you think is going to happen, and then usually they are going to ask you to justify. If you can justify with a legitimate biological reason for your prediction, you are going to earn points. Okay, so I just want to put that out there. But in this case, I've given you a specific example. So in normal hemoglobin, the 6th amino acid, which I showed you in that previous picture, let me see, I've lost my, is glutamic acid. So glutamic acid has a negative charge and is hydrophilic. When a point mutation occurs, valine is brought in at the 6th position instead of glutamic acid. Valine is hydrophobic. So predict the change on the structure and function of hemoglobin. So go ahead and put your answer in for C. What's going to happen to the structure and function with this change in amino acids? All right, so you guys are doing a great job again. Here's the thing that I would also stress, now that I'm kind of seeing your answers, it made me think, with predictions, a lot of times you just have to answer their question and make sure that you're answering all. So and is in bold, all the letters are capitalized, they're not bold. That means you have to address both of those things usually to earn a point. So this is a 4-point question. And so if you're predicting that there is going to be a change in the structure, and a lot of you told me, since glutamic acid was hydrophilic, it would have, positioned itself on the outer portion of hemoglobin. And since valine is hydrophobic, it's going to try to position itself in the interior of that molecule. So it's going to fold inward. So therefore the structure or the shape has changed, and the function of the hemoglobin will not be possible. And so some of you even went more specific and said it's not going to be able to carry oxygen as efficiently, and that's really good. So as long as you were stressing how that structure was going to change and that there would be a function, like less of a function of hemoglobin, you're on the right track. And then justify, so your prediction, justify your prediction for the effect of this substitution. And I'm going to go ahead. We've kind of already covered that. It kind of seems repetitive here. But essentially, what most of you have already said here, valine's hydrophobic. We know it doesn't interact well with water, so it's going to position itself on the interior of the molecule. Glutamic acid is hydrophilic. We know hydrophilic likes to interact with water, so it's going to be on the exterior of the molecule. And so therefore, that's going to cause a shape change and lead to hemoglobin no longer functioning correctly. So you would just kind of get into those specifics of, what the hydrophilic, what the hydrophobic is, and how that causes the folding to differ. So I do have the answer here. So if you are going to my slides, but I think it is about time for me to wrap up so you guys aren't stuck here all day with us. Here's a good picture of this. And you can see, I liked this one. So this was normal with that glutamic acid. And then with valine, you can see where that portion of the protein has folded inward because of that. So I think that's a really good visual. I've gone on, I talk about enzymes in a little bit more detail. I talk about, I've given a graph that you can analyze if you want to. And then I did just very quickly cover nucleic acid. So that'll be available to you if you do want to, practice some more about proteins. I've listed some different functions that we didn't get a chance to get into, but I think you guys are doing a great job, especially with your writing. So that looks great. And I think that's important. Remember on your test, 50% of your grade comes from the multiple choice and 50% from writing. So you do have to know what you're talking about and be able to express that well on your test. Thanks again for joining me. I think that's a big part of it, reviewing and challenging yourself to learn the material. So thanks again. And Sheri, do you feel like we need to take a break or just move on?
Wonderful. Thank you, Miss Cook. What we will do, I have a document to share with you for the next session. So I'll share that in your chat. If you could open it up and have that ready, we will take a 5-minute break, if that will help everyone to kind of get a little brain break for just a minute. So let me put that in there. I'm going to share to everyone. And then you can have that open. And okay, did that share? Okay, wonderful. Okay, so 5-minute break. We'll see you back in just a little bit. Okay, good morning. Miss Cook, can you see my screen?
Yes.
Thank you so much. Again, welcome to our second content review session at 11 o'clock. And this is, our focus will be on membranes. And excited to present today to our fabulous AP Biology students. And we will be looking at, of course, the phospholipid bilayer quite extensively. And Ms. Cook just really led us into this presentation by her discussion about proteins and how important and significant proteins are to the phospholipid bilayer. We'll be looking at membrane transport and how molecules travel across the cell membrane, aka plasma membrane. And it's exciting to see. Hopefully, I love graphics, so you'll see a lot of graphics. And in our first graphic, you see the phospholipid bilayer. You see a variety of other molecules that are attached to the bilayer, and we will talk about those specifically. I will begin with proteins as if where we left off, and we'll jump into some of our multiple choice questions to help us utilize the chat box and also our polling options for today. So again, let's jump into something that you've already taken a look at. And I'm going to stop my video just for a moment. But we are looking at graphic number 1, and this should look very familiar as it was the conclusion of Ms. Cook's presentation. And we see here that we have an embedded protein potassium channel. And our first graphic in our question, and I feel comfortable in going ahead and jumping into this because of our last presentation. So you're very familiar. I saw your notes on the structure of a protein. And in question 1, MaryBeth, if you could pull up our polling option and launch our polling for question number 1. And if you have any trouble with that, I'll be glad to launch it. But our question is, what protein secondary structure is part of the potassium channel protein shown above? So if we're looking at that graphic, we can determine, well, what is that structure? What are we looking at? How are those polypeptide chains interacting? What do we see? MaryBeth, let's see. Were you able to launch that? If not, I can launch it. Okay, so I have launched. If you could choose A, B, C, or D in your polling options.
Sorry, I forgot I had myself on mute.
Oh, that's fine. Miss Cook, I can't see them as I'm not sure why, but it's not allowing me to view their responses. So can you see that they are responding?
Yes, they are.
Okay, perfect. So if you could let me know at about a minute or so, or if we want to give them a minute and a half to respond.
Yeah, they've got about 15 seconds for a minute and a half.
Okay, thank you. I'm not sure why when your presentation, it would not allow me to see the responses as well.
Oh, really?
Yes, and that's why I was waiting. I thought, no one is responding.
Okay.
But you could see them. So if you can see them, that's fabulous.
Okay, so it is saying 66% of the students went with B.
Yes. Okay, correct. So let's talk about that a little bit and what we are seeing in the graphic. Okay, we know that the primary structure is the polypeptide chain. And then when we see, as we were described earlier, when the protein begins to fold and we see the coiled or helix shape of the protein, those are the alpha helices. And also they're the beta pleated sheets. However, in this question set, primarily we see the alpha helices. So I'm not sure in your polling which response you chose other than B.
We had 14% A and D and 6% C.
Okay, so a little bit of a mixed choice there, but the correct solution would be solution for answer B. Okay, let's take a look at our second question. And this involves quaternary structure. This particular question set came from, for teachers, the source was the MIT test prep. So I have that source at the top of this document if you would like to use that for question sets for students. So let's take a look at question number 2. And this question involves the quaternary structure of the protein. And does it have a quaternary structure? So will you launch that? I wish I could see the polling results, but Ms. Cook, if you could launch that, that would be great.
I've got it going.
Thank you.
How much time do you want them to have?
On this one, at least a minute.
Okay.
I'm going to move through this a little more quickly on this beginning section because the students had content review on this primarily in your session. I did want to start with it though, because the phospholipid bilayer is so impacted by proteins and their interaction and how the molecules will flow across the bilayer. So I did want to start with this as well.
Okay, guys, you got about 10 more seconds. Okay, so it looks like 53% went with C, but we also have A, B, and D selected.
Okay. Awesome. The correct answer is C. And teachers, I will send the solutions as well in a document to you, and it'll be available. So we have to remember that it is a quaternary structure because it has 4 interacting polypeptide chains. Now, if you look at the graphic, it kind of appears that these polypeptides are interacting with the neighboring polypeptides, and the overall shape is somewhat of a barrel, but it definitely is quaternary structure because you can see graphically it has at least 4 interacting polypeptide chains. So on that question, the best choice would be C. Now, what if you had to draw a cross section of where those where would the phospholipid bilayer heads look like? Where would they be? Typically, you would draw them onto the sides right here, because remember, if we look at this photo right here above our graphic, we can see where a protein channel may be embedded. So this represents something like this protein channel. So where would we draw the phospholipid heads and tails? Well, of course, we would put them right here to the side. So it's just an extension. So just a different way to look at this graphic to help you to kind of visualize, well, we see it, but where does it exactly fit into the phospholipid bilayer, and how will molecules travel across? Some proteins, are transport proteins, others are channel proteins. And we will look at a few more questions that will help us to kind of see the differences of those moving molecules. So I'm going to scroll down. We talked about our cross section for the protein channel. And now we're going to look at question 4. And this again was brought out wonderfully in our presentation prior about how important it is for those R groups and the interacting of those R groups to determine the structure of the protein. So if we're going to look at question number 4, what types of amino acids do you expect to find on the potassium channel polypeptides next to the tails of the membrane lipids? So let's take a moment and launch our poll.
I've got it up, I think.
Thank you. And remember what Ms. Cook said about how those molecules will interact depending upon whether they're near the tails or whether they're going to be near the heads of the phospholipid.
Okay, so we had 48% say B.
Okay, this was a little bit, I had several students when I gave this question, they said that as well, but the actual answer is D, nonpolar. Why do you think that it wouldn't be B? Think for just a minute and I'll look. For some reason, my chat box is not seeing my chat box, but why do you think that the correct answer would not be B? If you selected D. Okay. There it is. Okay, I don't see any responses, Ms. Cook. Okay, here we go. All right, awesome. Okay, nonpolar molecules aren't negative. Polar is a negative. Okay, so Megan, you made a good, your thinking is wonderful there. What I want you to remember is that, remember those charges are not they're distributed equally. So a nonpolar molecule has no separation of charge. So no positive or negative poles are formed. So in essence, we can just say nonpolar, but not necessarily negatively charged. The electrical charges of nonpolar molecules are evenly distributed across the molecule. So a little tricky question, but I think it brings out a good point. So keep that in mind when you're thinking about polar versus nonpolar. Okay, let's take a look at question number 5. And you're doing wonderful. Let's pull question number 5.
Okay, it's up.
Okay, great. Now we're talking about polypeptides next to the heads of the membrane lipids. I can see chat, but not the poll.
Yeah, I don't know why you're not able to.
I don't know. I couldn't in your presentation either, my friend. And I thought, why are they not answering? But they did wonderfully.
Okay, we got about 10 seconds for a minute.
Okay, great.
Okay, so we had kind of mainly torn between two. So 68% at C, 21% at A.
Okay, it is actually A. Okay, the reason why it is A, of course, polar probably stands out quite a bit. But remember, in polar molecules, there is what? Is there an even distribution or an uneven distribution of charge? So we would have to say A, positively charged, because there's an uneven distribution of charges. Kind of the opposite of what a nonpolar molecule is. So something to just bring out and make you think about a little bit more. So the answer to that one would be A, polar, positively charged. It can have a positive or negative charge, but in this case, because it's polar, it's positively charged. And again, in our prior question, it was nonpolar, but because the distribution of charges are even, we're not going to select positive or negative as being the primary charge. Okay, so great job, guys. All right, just something to help you think a little bit more about polar and nonpolar charges. Okay, so now let's move down a little bit more into this graphic of cell membranes and how they're constructed and some of the wonderful molecules that are a part of the cell membrane and why Ms. Cook's presentation was so important, because we have to have that background and that foundation to really understand the cell plasma membrane. Why is it important? Well, it's used for protection of the cell, for transportation for the cell, and communication in the cell. And I don't know where you are in your content at this point, but we know that cell-to-cell communication is a big part of this course and how it interacts with all of the different units. Well, we see it again here and how important the cell membrane is in that interaction. So what are some of the awesome molecules that Ms. Cook told you about? Well, we talked about phospholipids, proteins, carbohydrates, and we also talked about cholesterol, which I'm not sure on this graphic. They have it over on this side, I think primarily because of the communication part of it. But we know that that is embedded into the phospholipid bilayer, not in between here, but actually embedded in this part of the phospholipid layer. It can be embedded here or below, but we also have these very important channel proteins that we were talking about. And again, how they impact how molecules will flow across the cell membrane. And we'll be getting into transport in just a minute. Okay, so let's look at question number 6. I like to ask you a lot of questions because it helps us to kind of think through why one answer might be more applicable to what we're doing. So take a moment and let's poll question number 6. And you could look at the graphic. That will help you to respond.
Okay, we got about 5 minutes for a minute and a half, or
Okay.
5 seconds.
All right, that's fine. Thank you.
Okay, so we're almost evenly torn between D and A. So 41% with D, 38% with A.
Okay, the solution is D. That is correct. So let's look at A. Why would A possibly not be correct? What is a key word? A phospholipid bilayer with embedded what? Carbohydrates. Do we see that the carbohydrates, are they embedded in the phospholipid bilayer, or where are they positioned on the phospholipid bilayer? So that's a key reference to help us to determine between A and D in this question. We can see that we have, in this graphic, carbohydrates here, here, here. But are they embedded like some of the other molecules, like the protein here or the cholesterol molecule? What do we see? And sometimes I can see where you might think this right here in this particular graphic might look like a carbohydrate. Okay, so let's keep moving on. Now we're getting into some really important parts about the phospholipid bilayer and how it influences selective permeability. So a couple of things, and you will not have time to do this on your test, but what I have my students do, and it really helps them to take maybe a difficult passage or a passage with a lot of words and break it down. I call it a shrinking summary. So if you practice this, it's really helpful in helping students to find what is the most important thing that they're reading in the passage. And sometimes on your test, you're not going to have time. As Ms. Cook said, you have about a minute and a half. But if you practice this tool outside of just test taking, it will help you to read more fluently and to understand, to comprehend what you're reading. So I've demonstrated it in this passage. It's called Shrinking Summary. It's where you take and break down the paragraph into 15 words, and then you break it down into 7, and then you break it down into the most important word. So if you can see, Ms. Cook, can they see the shrinking summary comment over to the side?
Yes.
Okay, so I want you to practice this. I've given you a model and an example, but I just want you to try it with this passage. So I'm going to give you just a couple minutes to do that. And I want you to take what is highlighted and break it down into 15 words. My example is membranes, selective, permeability, small, nonpolar molecules, freely, large, polar, move, channel, transport proteins, water through. Okay, so take just a minute and try that. in the chat if you would share someone share their 15 words or someone share their 7 words, whatever you feel comfortable sharing. Preferably not your 1 word, although that is very awesome because a lot of times students will share the exact same word in a passage. Sometimes I'll see the same 7 words. So if you would go to your chat and share what you feel your most important 15 words are, or your most important 7, that would be fabulous. Great. Jasmine, awesome, has shared membranes, semi-permeability, structure, fluid, hydrophilic transport. Wonderful. Remember, your words need to come from the passage that you're reading. And if you practice this enough, it's called shrinking summary, you will be surprised how quickly you can read and how you're highlighting the significant, most important concepts of the passage. Great. Megan, membrane, selective, hydrophilic, nonpolar, molecules, embedded channels. Excellent. Good job, guys. I know it's a little different if you've never done this before, but it's just a strategy to help with comprehension. And with some of the tier 3 vocab words that we have to have in our learning for science. Great job. Good, Brennan. Good. Thank you for participating. Awesome. Okay, so now back to a question. Let's take a look at this graphic. Okay, and you'll see a lot of these on AP exam tests, questions like this that are applicable. They want you to think about a scenario or a situation. So in this graphic, we have a tube, and representing our phospholipid bilayer is this line right here. So we're going to have fluid that's flowing from this side to this side, back and forth. So what are we focusing on moving across this membrane in this graphic, in this question? Well, we're looking at what? So in just a moment, type in the chat what you know about any of the words or the graphic that you're looking at. So what molecule are we focused upon right now? What's our molecule of choice that we're looking at in this picture? So just type that in the chat. It ends in what? Yes, glucose and fructose. Excellent, thank you. Now, what monomers? We know that glucose is the monomer of what larger molecule? We just learned this too a minute ago. Great. Carbohydrates. Thank you, Riley. Awesome. Perfect. Okay, so let's take a look at this question. All right. First of all, on side B, we have how many moles of glucose? What are we looking at? 2.0. And then on side A, 1.0. So which side has the greater amount of glucose? Well, obviously side B, and it's the opposite for sucrose. Okay, so it says the drawing above shows 2 solutions of glucose and sucrose in a U-tube containing a semi-permeable membrane which allows the passage of sugars. Which of the following accurately describes what will take place next? Okay, so take just a minute. Think about that. And also think about how substances move down their concentration gradient until their concentration is equal on either side of a membrane. So let's think about that. So which direction will glucose move if it's moving down its concentration gradient? Which direction will sucrose move if it's moving down its concentration gradient? And I'm sorry, Ms. Cook, we need to launch a poll.
I did. We're at a 106.
Okay, thanks.
Take maybe 10 more seconds. All right, so we had 50% go with D and 25% with C.
Okay, so actually the correct answer is D, so good job. Let's see why C would be incorrect, okay? When we're looking at the tube, we know that molecules will move down their concentration gradient, and I have a graphic for you. I probably should have used this question a little bit later on as we build to this, but I wanted to see how you would do, and you did pretty well. Good. So if you're looking at side B, we know there's going to be more glucose on this side. So which direction is it going to move? It's going to move from B to A. If we're looking at sucrose, which direction will it move? From A to B, because there's more sucrose on this side and there's more glucose on this side. So C, no net movement of molecules. There will be movement of the glucose and the sucrose because they will be moving down their concentration gradient. Okay. So let's look, let's see, I'm trying to decide if I want to go ahead and do number 8 or number 9 because our time goes by so quickly. Let's go ahead and do number 8, and then you can refer to number 9 a little bit later on because I have a few more graphics that I want to show you just to make sure. It looks like we're 50-50. And I have a graphic that I think will help us a little bit better to review this movement of molecules across the membrane. So let's try number 8. And we'll launch our poll in just a minute. Look at the same figure, but let's assume the membrane is not freely permeable to both glucose and sucrose, but only to the smaller of the two sugars. Okay, so in this question, first you have to think about, well, which sugar is smaller? Which sugar is smaller? So we have to think about that. And then that is the one that we're going to focus on in our question. Now, what results would you predict? So let's launch our poll.
It's up.
And if we know that glucose is the smaller sugar, and how do we know that? Actually, in this question, it gives us the ratio of carbon to hydrogen to oxygen. And we can see that glucose would definitely be a smaller molecule, even if we didn't know the structure of what glucose looked like or sucrose per se.
Oh, I wasn't paying attention to the time this time.
Well, that's okay. That's okay. And you know, these are applicable questions, so we have to really think through them sometimes. And they're going on their second hour online, so I'm really impressed. They're doing a good job.
All right, so we had 63% go with B and 26% with A.
Yes, the correct answer is B. Okay, so like I said before, first you must know that glucose is the smaller molecule and only focus on its concentration. Because its highest concentration is on side B, it will move to side A. Okay, fabulous. All right, so this helps me to know, I feel like, you know, most of you did get that. That was very, very good. But I'm going to scroll through this just a little bit, move down to this graphic, to graphic number 4. Okay, so we can have a little bit better review of how molecules are being transported and how important it is to see the different ways. If it's simple diffusion, if it's facilitated diffusion, if it's active transport. And if you've had this content, you probably remember those ways and those methods of transport. So let's look at graphic number 4, and then let's take a look at question number 10. Which figure represents active transport? Let's see, I don't have a poll set for that, but we can use our chat box. And in this graphic, you have the choice between number 1, number 2, number 3, and number 4. So just looking at this graphic, what do you think in the chat box? And how do you know? What are 2 ways that you can tell that you know for sure that it's 1, 2, 3, or 4? So in your chat, you will respond with 1, 2, 3, or 4. And then if you could elaborate a little bit and tell us, how do you know that? Ah, okay, good job, guys. Okay, it looks like you've seen this graphic maybe before. So it probably would have been a little better for me to start with this one. But it's helpful for me if we jump in and I see where you are. Okay, so yes, it should be number 4. And Sawyer says it uses ATP and it is against the concentration gradient. Excellent, good job. Okay, so let's see. What about question number 11? Just to make sure we clarify this a little bit. Question number 11. Which section shows simple diffusion? Answer that. Put that in the chat box, please. Simple diffusion. Ah, nice job. Good. Good, Allie. Amanda. Awesome. It's number 1. Okay, so this is familiar. This is easy breezy for you guys. Good. Okay, let's go ahead and do the last 2. Question number 12. Which section shows facilitated diffusion with a carrier protein? With a carrier protein. And then 13 is going to be with a channel protein. So you want to think about which one, carrier versus channel. Excellent. Good job, guys. Okay, so number 3 is carrier protein. Good. And number 2 would be channel protein. Excellent. So you can recognize what proteins, what they're doing, how molecules are moving with their concentration gradient or against their concentration gradient. Very good. So The other question, the first question that we tackled, it does require a little bit of review on this prior. So excellent. Good. Okay, so I'm going to go back to this question up here and let's finish number 9. Okay. Number 9. Let's see if we can do this one. Look at the same figure and we're looking at this figure right here. Okay, we're still back to this question. Because you can, these are pretty common. Miss Cook is an AP reader, but sometimes, you know, I think she would maybe, these can be common multiple choice questions for you. She can let us know. Okay, so we're looking at the figure and assume again the membrane is not freely permeable to both glucose and sucrose, but only to the smaller of the two sugars. Okay, so which sugar are we thinking about? We're thinking about glucose. What effect will the movement of the sugar have on the water level in the tubes? Okay, we haven't talked about water in this particular example, but let's think about that for a minute. Let's see where we are on water. Think about water. This one always tricks my students.
Okay, so we've got 57% at A and 24% with C.
Okay, excellent. The correct response is A. Good. The water level will go up inside A. Okay, so we have to think about a couple of things. First of all, you probably remember terms like solute. When glucose, when it moves to side A, what's gonna happen? That means that the total amount of solute, the glucose on that side, is going to increase. And that is going to make that side hypertonic. Okay, what happens with water in a hypertonic situation? What does the water, can you, in the chat box, can you tell me, what do you know about the movement of water? Okay, good. Sawyer, see, water moves hypo to hyper. More water flows in, yes. Hypo. Water moves to where there is a higher concentration of solute, yes. Good. Okay, good. Thank you for participating. Yes. Good. And we're going to talk about, if I can squeeze it in 10 whole minutes, a little bit about water potential. And there's so much that I'm not going to be able to do, but I'm going to try to get in as much as I can. So excellent. Okay, so let's think about water a little bit more. Let's go to, I'm going to scroll down, and these questions, you can work through these in your study time. I will give solutions to your teachers so that they can look over this and maybe review. You can review on your own. Let's see, which one do I want to do? This is active transport. This is a great graphic as well. I have on your notes, remember, water moves from a region where water potential is high to a region where water potential is low. We haven't really addressed, also think of water potential as potential energy. So let's look at a water potential problem. Now, my kids love these problems. So if you have just a moment, let's remember in this formula, you're going to have an ionization constant. Typically, I don't know, Ms. Cook, we've always had salts or sugars, ionization constant of 1 or 2 in our problem sets. We're going to talk about the molar concentration, pressure constant, and temperature in Kelvin. And you have to do a conversion for the temperature change if the temperature is in Celsius. So my kids love these problems, but typically on the exam, they will forget to add the negative sign. Okay, so something to always remember when you're working out these problems. So let's try, if you have your calculators available, let's just pull in one of these math questions and we'll see how we do. So here we go. Can you see this? Do I need to, if I do this, let's see, does that make it bigger, Ms. Cook?
Yes.
Okay, so we have potato cores, and keep in mind on your AP exam, these formulas are completely available to you. You don't have to memorize anything. You just have to apply and think about what the question is asking you to solve for. So this question, potato cores were placed in solutions of varying concentrations and were found to neither gain nor lose mass in a sucrose solution of 0.32 moles. Use this information to calculate the solute potential of the potato cores. The temperature of the solution is 22 degrees Celsius. So, what is the first thing that you think you need to do? I usually have my kids go ahead and convert the temperature change. So, what in the chat box, what would the temperature change be? What would it be if we need to convert temperature in Kelvin from Celsius to Kelvin? Could you put that in the chat box, please? Good. Oh, these are math kiddos. This is wonderful. Okay, great job. All right, so we know that. Okay, so now what are we going to do? We're going to follow this formula up here. What's something that kids forget? This little sign right here. Okay, so what is the ionization constant? What molecule are we talking about? If you would put in the chat box, what would the ionize? Okay, good, Sawyer. Good, Riley. Talking about sucrose. So what is the ionization constant going to be? Is it going to be 1 or is it going to be 2? Good. Excellent. What is a typically sodium chloride? Sometimes I think that's used in some of the questions. The ionization constant of a molecule of a compound like that, what would that be? That would be what? It would be 2. But in this case, we're talking about sucrose. So now what do we have to do? We have to find what? The molar concentration? Does it give us the molar concentration? Yes. Okay, so we're going to multiply that. It also gives us the pressure constant. So we will multiply that times the temperature. So what is the solution to this question? Question number 17. If you could place that in the chat. Fabulous. And my kiddos love. And again, I will have this all written out, all of the solutions worked out for you and send that so that you can see as well. Okay, so good job. All right, so we're continuing on with this thinking of water potential and how special and wonderful water is. So if we look at question number 18, and we have an entire 5 minutes left to end our day. So we will probably end on this question. There's so much I would like to get through, looking at graphene and a lot of other things, but let's try this with water potential. So again, the formulas are given to us here. We know that water potential equals pressure potential plus solute potential. Okay, that was a given. So we're thinking about that and we're looking at this question. In beaker B, shown in figure 4.4, what is the water potential of the distilled water in the beaker and of the beet core? Now, typically my kids do an experiment like this. Sometimes potatoes, carrots, it just depends upon the choice of that year. But I want you to try this question and we will launch a poll and talk a little bit about it. I want you to think about, remember, water potential equals solute potential plus pressure potential.
Okay, we'll try to take about 13 more seconds.
Yes, and then I think, Ms. Cook, we only have time for one more question to wrap up our day.
Okay, so the results on this, we had 50% choose B and 29% choose C.
Okay, the solution is B. The correct response is B. The water potential of distilled water in a container open to the environment is 0. The water potential of the beet core is minus 0.2. Since water potential equals solute potential, water potential of the beet core is minus 0.2. Okay, so let's try number 19. And thank you for reviewing questions today. Some of them are challenging, but it's always good to challenge yourself. It helps you to see what you might need to work on and focus on in the future. So number 19, let's try it. Which of the statements is true for diagram 4.4?
Okay, so about 13 more seconds. Okay, so we're kind of divided. We had 32% with A, 23% B, 18% C, and 27% D.
Okay, so this is one that we would want to spend a little more time on. The solution is A. The water potential for both the distilled water in beak core and pressure in beaker A is 0. In choice C, the beak core in B had a pressure of 0.2, whereas in beak core in A is 0.4. So the B in B would be less targeted. So that, and again, I would love to continue on. There's so many good questions in this review, but we will conclude for today. And thank you so much for participating. Ms. Cook and I really appreciate it. We wish you well. We know you're going to do wonderful, spectacular on your AP exams. I have an engagement tool that I use, and I don't know if you've used the Wheel of Names as well, but it's really helpful. We have fun with it in our classes. I'll launch it, and you can respond in the chat. Here are all of the different schools that are involved right now in our AP biology cohort. And we just thank you so much for participating. Question is, if there's someone here from E-Town, do we have any E-Town participants? This is a fun wheel to use, and E-Town, if you're here, we're just so thankful for you and your participation today. We're thankful for every single county that's here today. So that concludes our review session. I'm going to stop sharing. And Ms. Cook, did you have anything else for these amazing students before we close?
No, thanks for joining us, and hope you guys have a good day. Continue the good work. You're doing a great job.
Okay, great. I'll stop recording. Have a good one. Bye-bye, guys.
Bye.